So let's say you've just opened MATLAB. You've run your max GPU all to make sure you have your CUDA code running. And now you have kilosort in your path, and you can check that it's in your path by just going here. You have to make sure the kilosort you have is the one in your own folder. Right? This is going to be even more important when you're each going to start changing your own local copy because you're trying to do something. Right? You don't want to run some other random copy. You want to run your own copy. So you have to make sure it's right there. And then this NPY MATLAB repository has to be there so we can save data for phi for the manual GUI where we do proper data visualization. Okay, so it looks like I'm set up correctly because I've logged in as guest 23. And so now I can run kilosort.
Do you have to run the max GPU every time you...
No, just the one time. Yeah, and it compiles these like binary, CUDA files underneath, and then it's able to use them throughout. Okay, now the first thing that happened here, notice that when I started, it already filled up these things for me. So that's because I had started it before and ran through it. If you're starting the first time, you should see just ... in all three of these folders. So that's like a clean, nothing should be filled out here. That will be like a clean initialization of the GUI. Because GUI is trying to remembering this thing you did last time, so it loads it for you. But the first time you run this, it should be completely clean. That way you know that you're really running your own new copy, not some other random one from the server. Okay, and so what you do is you'd select data file, and again, you want to select your own copy of data. So I've gone up all the way to hackathon. So just to be clear, the whole structure here of like the entire Genelia server, right, is there's this kind of file server up here, and then everyone's in groups, including the labs, and then we are in hackathon. And so in hackathon, there's just the name for it. We go to data, and each of you has made a folder there, and I've made one in guest 23, and I'm going to go into guest 20. I'm going to go in guest 23, example data. And now, okay, because I've already run it, there's all sorts of output files here already. But you should only have this one in there, bin. Or maybe you'll have the settings as well. But definitely the ap.bin file, so this is the big one. That's what you want to load into a C2P GUI. Now, the kilosort GUI. And now those of you who have your data in a different format that you'll have to convert it, right? So we'll work through that tomorrow. But everything has to come through as a binary file in the end. Okay, so as soon as I loaded it, you should see that the data kind of popping up on the right here. Now, this little GUI that we made this to run kilosort has a few purposes. One of them is just to check that kilosort detects the channel kind of order of your data correctly, and the number of channels, that basically it's not messing up on the reading side. So if the data looks good on the right here, it means it's correctly identified. Things like, the number of channels comes here, 385. And I don't know, if you look at the very first time that you load this, it's telling you that, it's going to guess the number of channels. And what that means is that it knows it should be a number pretty close to 384, because that's how many channels are on the NeuralPixels probe. But it also knows that people sometimes add an extra channel for like synchronization purposes. And so that's why it's 385 here. And it can guess that because the number of samples in the binary file is a multiple of 385, but it's not a multiple of 384. And so if you have something like, yeah, go ahead.
I was going to ask, so mine guessed it was 410. It's the same file, and it's the same road layout, and all the same parameters.
The exact same file.
Yeah.
It's very strange. Can you change it to 385?
As soon as it just finished saving, yeah.
So it won't let you change it to a file that's not a multiple of 385.
Okay. No, it won't let me change it. It says guessing that the number of channels is 410.
Okay.
If it doesn't look right, consider trying 410 or 416.
But if you just type it by hand, if you type by hand 385 in there?
Yeah, it auto-corrects back to
Okay.
410.
I'm going to just make a guess here that you were copying this file and somehow it got interrupted and it didn't copy all the samples. And maybe just start over and copy that file over. Anyone else has this problem? No? You do?
It allowed me to change it, but it guessed the wrong
That's funny. It should always guess the same, but you know, like humans, we sometimes make different guesses. It's just okay. Well, so assuming you can all change it to 385, as soon as you do that, you know the data on the right should now look correct. I mean, you'll see that it looks better if you zoom in more. So to zoom in, press Shift and kind of zoom in. Shift, zoom in. Now, I have a mouse, so it's easy for me. There's one more mouse for whoever gets there first, but I'm just kidding. But it's easier with a mouse, of course. So press Shift and zoom in. Now, if the data wasn't loaded correctly, you might see something very stripy like that. So if, for example, the number of channels off by 1, let's say, then on every row, it will just keep getting shifted one further. And it's going to have like a stripy artifactual pattern. But it's not, right? So like maybe we zoom in even more here. So I do this by holding Control. Now it's zooming in on the y-axis. And so, you know, this lets us kind of see all the spikes in here, right? It's very nice, very big spikes. And this is just on the raw data. There's no processing whatsoever. So in red is going to be the negativity, and in black is actually the positivity. So all of these are going down and back up. They look like little, okay, what I like to think is they look like little, I don't know, like fish in an aquarium or something, right? There's like they're like clownfish, right? They're red and blue. So that's how you parse them apart, by just parsing the little fish. Anyway, so we can see the spikes. Okay, we get an idea. So now we you know, if we can see spikes and they look like there are many channels consecutively, then we kind of already know that it must have loaded the data correctly. Okay, and so the scroll wheel alone is just going to scroll you through the data, left, right. Look at all these beautiful spikes that Susu collected. Where would this be, Susu? What brain area? Where's Susu? This is the thalamus? Okay, so I guess, yeah. So it's big firing rates, right? Lots of overlaps. There's a lot of, this is going to be, you know, this is going to make spike sorting hard because we can see lots of situations where spikes are overlapping. So we'll go through that and we'll see how well ks2 can do in this situation. And I guess if that's the thalamus, then if I zoom out, at the top where there's no spikes, I guess that's got to be cortex then. Okay, so let's try to find some spikes up at the top. I'm not sure if I'm zooming correctly to the top, but I can try. Okay, so controls will also tell you how to zoom in correctly through these things. So zoom, scale, move. Scroll and Alt, Control, Shift, scroll. Okay.
Is that the right file?
Disable nearest channel. Okay. Okay, one more thing that you can do, right? So this is made as an image. Oh, I see. If I click, it's going to take me up there. Okay, so if I click on this picture, it shifts my center of focus to that part of the array. In this case, it put me at the top where we can see the spikes are much smaller and less frequent. And then every time I click, I guess it also moves me right because I'm clicking to the right or I'm clicking to the left. So that's something nice. So this whole GUI was built by Nick Steinmetz, who, you know, recognized that we should have an easy way to look through the data in here before we even run ks2, just to get a sense of what's going on. Okay, zooming back out. I think I've exhausted the things to do before I run something. You can jump through the recording, I guess, at the bottom, if you'd like. You can zoom in on the channels. Okay, so this channel map is completely specified by kind of a few parameters. You'll have to build your own for your own probe if you don't have it, so we'll go through that together for those of you who still need to build a channel map. But if you like say, so here when you select the probe layout, if you say new, basically you're going to need this information. You're You're going to give it a name. You're going to give it some x coordinates for each site, so in order. So your sites are in the data file, your sites are coming in some usually scrambled order, like 23, 1, 12, etc. So make sure you have the x coordinates in that order and the y coordinates in that order. Then you can also make multiple like shanks if you have such a probe. And then, so that also has to be one number for every channel in that order. And then the channel map, so by default, this could just be 1 through your number of channels. But if it's like, if you want to skip some channels because they're dead or something, you could in principle skip that, but it's probably best to just say 1 through the number of channels. You can also put expressions in here. So for example, I can say 1 through 32. And then maybe for x coordinates, maybe I have like a linear probe vertically. So the x coordinates should be like zeros for all the sites. So I could do this. I guess they should all be horizontal vectors. And then the y coordinates then are the ones that are changing. So they will start with 0, and maybe I guess it will be like every, let's say I have a 20 micron spacing between my sites. And so 20 to 20 times 32. Maybe start at 20, that should be the right number of sites. Okay, so I guess I could have just specified that more neatly if I just said 20 times 1 through 32. Okay. Shank index, let's just say they're all on the same shank. So again, it's like ones of 1 to 32. And that's it, blank for none. Okay, so I just made a new channel map for a 32 linear channel probe. Okay, it can get more complicated, but that's just to give you a sense of what it is and make sure that you have that information. Yeah?
Does the shank index still work?
It works for display purposes. It does not really help inside of kilosort. And we can, I guess, if that is causing issues, we can probably add that pretty easily right now. I mean, during this workshop. All right. So now going through these parameters, number of channels, we talked about that. Your sampling frequency, make sure that's correct. And then your time range. So the thing, you can either sort the whole thing, or maybe you just want to like do a little sort to test something out. So you could just say, I want to sort the first 600 seconds of this. So that'll be like 10 minutes. Oh, maybe that's still too much. Let's sort the first 200 seconds. That should just go very quickly by, and you can test if something failed or things like that. So that's what you can, how you can use time range. Okay, now the first thing that happens in kilosort is it's going to discard any channels below the minimum firing rate. And so it estimates, the very first thing it ever does is it kind of estimates using kind of rudimentary kind of spike extraction methods, just if there's activity on some channels or not. And then discards all the channels that it estimates not to have activity. If you didn't want it to do that, you could just set this to 0, and that will include all channels. Or if you want to set the threshold higher or lower, you can do that too. So let's just say we leave it to 0. So these are the 2 thresholds. So these are actually now the 3 parameters for the actual algorithm. So those are the 3 that at the end of the talk I described. The first one is the threshold for the kind of optimization pass, the forward pass. And the second one is the threshold on the extraction pass, which is much lower, just to make sure we grab everything because we want to set an adaptive threshold for each neuron. So we must first make sure we have more spikes than we need to cut out, however many of them we want to. Again, these default parameters, you shouldn't, one thing that's probably not a good idea is to kind of randomly change them and expect things to change. So what we should do is we should run default parameters, get an idea of how it fails, and kind of understand how it fails. Maybe like neurons are kind of crashing into the noise floor, so then we'll think, maybe we have to lower these thresholds even more, or maybe we're getting too much noise into our clusters, and then we want to lift the threshold. Kind of that. Those are the kinds of changes we want to make. All right, so I've already told you about these, so let's just leave them. We'll modify them only if we need to. There's also a way to see advanced options, which is to just type them into the keyboard. But again, there's nothing like that's going to super change the results of the algorithm. So there's no like magical parameter you might be able to set. There's relatively few parameters. So if something isn't working, we might have to dig in a little bit deeper to see if we can add some extra pre-processing of the data or post-processing or whatever. Just make it work for the kind of very variable data sets you guys all have. Okay, and if you've done that, and then at the GUI here you can run things in stages, and I recommend doing that just because again we can inspect the output of every stage. So I just pressed pre-process now, and it's pre-processing. Okay, looks like it's, yeah, okay. So we can now wait. So you've probably pressed run all, which will run all the different steps. And you'll basically still be done pre-processing. Good. Okay. But this is not responsive anymore for some reason. So what I would have wanted to show you is basically after the pre-processing is complete, you can see like the filter data, and that again gives you, so that's one step of pre-processing is to kind of high-pass filter basically the data. And so if you see your data high-pass filtered, you're one step closer to what Kilosort is actually going to use for spike sorting. So it's good to see that one too. But for some reason, it's completely stopped there. All right, well, I'm just going to press spike sort then. It seems like that's working fine. So that's pre-clustering to reorder batches by depth. Okay, so that's, now this is only going to have 200 seconds worth of data here. Now, if you look at the command line, you can always see what Kilosort is doing underneath the hood. And so it's the first figure it threw out is this figure of kind of batch-to-batch correlation or distance. Okay, looks like the graphics are a bit messy when it runs, but okay, basically now because I'm only spike sorting 200 seconds, there was really no time to have any kind of drift at all. But if you guys have sorted the whole thing, then you would have gotten a picture with maybe more stuff in it. Maybe I see one of these pictures. Does anyone have a picture? Yeah. That's it. All right. So there's still not much in this, right? Because Suzu's recording it, this one was particularly stable. Whoa, look at all those merges it's doing. Okay, so you want to sort the longer segment of data just to, usually it should not have to do that many merges, but it might have to do with us sorting a relatively small portion of data. Okay, so it's merging, it's splitting, that's okay. Okay, what I'm not going to do, so I've already kind of run this once, and I've downloaded the results to my local computer so I can show you those in file. So that's what you would do from here. Well, the first thing is you do, there's this other plot that might be good to understand. This other plot, what it does is, so for every unit, they get sorted by their spatial location, and so that's why the spatial components all look like that, kind of arranged in a line. So we can tell from this line, for example, if you look over here, if you can see anything, that it skips some channels, right? Presumably because those channels didn't really have much activity, so there weren't really any units found there. So it kind of skipped next to the next set of good channels and continued that way. Okay, so those are the spatial components. And there we go. Okay, and there's also the temporal components. And what these are, you could think of them as the maybe like the component on the biggest channel. It's not quite that. So it's not quite the time course on the biggest channel because they're obtained by this SVD procedure. So they're the first, they're the first SVD component of the entire waveform. So whatever is the biggest time course in your waveform, that's what's plotted there. And they're all nicely aligned to sample 21, which you can see here. And then, you can see some patterns. For example, kind of in this middle area, a lot of neurons have kind of a more multiphasic, perhaps, waveform, because there's this initial part up here, and then all of these other ones don't, like here and here.
So it's aligned to sample 21 because that's
Maybe
how
that's
it's defined.
noise. It's probably noise. That's by construction. You can change which sample it aligns to, but by default it picks like 61 samples, and the most negative peak is at 21.
What is the sampling frequency with the answer?
30 kilohertz.
So it's 2 milliseconds.
Yeah. Yeah. Yeah, and actually, so these things that I pointed before, I think those are going to be noise. That's why they have this extra stuff in there. So we'll look at those more closely in the phi GUI. The other plots are going to be the unit amplitudes. Again, not in nice millivolts units, because we are in normalized units, because there's all this kind of spatial whitening happening on the channels, like I guess I didn't really mention. So there's not just high-pass filtering down on the channels, there's also spatial whitening, which is like, it's exactly like high-pass filtering, but across channels. So this is kind of trying to remove these correlations that kind of noise has. Not noise, not electrical noise, but like actual kind of background noise in the tissue from the activity of lots of neurons that usually has activity that's kind of spread out over many channels. So we kind of want to make sure we don't identify that as a spike. So that's what the channel whitening operation does. So it's a little bit like subtracting from every one of your channels the mean of nearby channels. It's not quite that, but it's a little bit like that. Just in the same way that high-pass filtering is a little bit like subtracting the mean of your time points all around your time point.
Is it doing common average referencing?
I think by default it is, but we'd have to check inside the code. But I think by default it's doing that as the very first step before any high-pass filtering. Okay, what are we looking at here? Unit amplitudes, okay, and as I told you, they're in units of this threshold that we're setting. So again, this is template units, not millivolts. And so, anything around 20 or so is really good. You won't really have many things above 10, so below 10. If you do, there'll be very few of them, roughly speaking. And now this scatter plot tells us spike count versus amplitude. So, there's a whole distribution of them, as you might expect for real neurons, but also probably a lot of these with low spike counts are more like noise and not really neurons. Probably would get rid of them if we ran a longer thing, or maybe not, I don't know. We'll have to check it out. Okay, so that's what these plots are. Any questions about these 4 plots? They just exist there to give you a bit of a quick feedback of how you're doing, right? Give you a quick sense of what's in the data, if the optimization is working. It's a quick check on that. Okay, so when this finished, okay, so the spike sort step finished, and so the next step, the next button that activated here is save for phi. And so it's saving the data, and that's the last step. And now you can load it up in phi to get access to a lot more visualizations of it, and that's what we will do next. There we go. Okay, so I have the filter data here. I didn't even have to press preprocess for it, because the first thing it does is computes a whitening filter, so it can show me what the process data would look like. And so the way in which this differs from the raw data is it's high-pass filtered and then channel whitened. So it will look a lot more like sparse, because we've presumably removed all of the slow fluctuations in time and space. And So now if you zoom in on some of these, right, they'll
Quick
look.
question, how are you toggling between the filtered and the raw?
Oh, I pressed 2, 1 and 2. So if you go to controls, it tells you that 1, 2, 3, 4 enable different data views. And so, assuming it's loaded, assuming you could see the raw data, you should be able to see the filtered too. And then you can go back and forth. But then mine, for example, just froze. There it's back. Okay. So let's go back to our good spikes down here. Maybe zoom out a little bit, back to filtered. Right, so you can see that if I toggle back and forth, you know, kind of quickly between these two, you see how like a lot of the stuff on many channels has been now kind of focused on just those few channels. And believe it or not, that really helps you identify. So you can identify the spikes much better from this than from this. Right, so basically what looks like is happening, if you look at it closely, is all these like fields kind of extended over many channels of every spike get kind of canceled out. And only like the big parts on the central channels is really the stuff that's discriminative, that's very different from spike to spike. And that is the part that we're going to use to do the spike sorting. Okay, so just keep that in mind. If your filter data looks like this, it's actually a good thing. That's good. There's all these like very different patterns on these spikes. All right. Next step is to load phi. Should we go through that as well now? Yes? Good. You guys all ready to install phi on your local computers? Okay, so
I couldn't save the data
what did it say?
for phi. It says undefined function write NPY.
Okay, so you also need to add to your path the mpy-matlab repository. So if you go to the instructions, you can clone it from right here. So clone that and add it to your path. So that's basically just a little set of small functions. All it does is it takes a MATLAB array and writes it out as a NumPy array for Python. Because phi is a Python, it's completely based on Python, so it needs these data file inputs. Okay, so now what you guys are going to be doing is, so forget about no machine for a moment. Just minimize it, put it away somewhere. And the next step now is to install phi. So search GitHub phi, or take the link from the instructions, but you should be able to find GitHub phi on the Cortex Lab website. And if you go down, it will tell you that the installation instructions are in this document. And so probably the best way to install it is run these commands. Now, you need Anaconda on your local computers. That's really the first step. And that will be the biggest thing you have to download. So if you don't have Anaconda, then you just click on that. You'll want the Python 3.7 version, right? 2.7 is no longer really anyone using it. You want to check it for your operating system, Windows or Mac OS. You download it and you install it. And it's a pretty big download. Let's see. So I already have it, so I don't have to install it. It's like okay, it should be very fast. It just did it for me. And
You can probably also go for Miniconda, it's much smaller.
yeah, but it might not have some of the requirements. And I've had issues with that before. Okay, so please install Anaconda. And if you have installed Anaconda, then you will be able to start an Anaconda prompt. And what that is it starts up and gives you all the paths to all the Python code and environments. And in particular, it allows you to run the commands which the commands which Cyril has in his documentation here, which you'll have to run one by one. Okay, you guys can, assuming that you did all of that, you should be able to cd to the directory where you just processed data on the server, and you should be able to start phi there, and it will look like this. Okay. So let's all try to get there. I'm going to leave open, I guess, these installation instructions. But I mean, you should here. So I'm going to start going through these, and you guys can either follow along or just look here, and you'll get a chance to inspect on your own later on. Okay, so there's all these views around, and they're nicely arranged. You can drag them. I don't know if they scale correctly on your monitor. I guess usually you want a big screen to be able to see lots of these views very closely, and you can. I don't want to mess it up because it's arranged so nicely, but you can take this out. You can take it out and you can put it somewhere else. I'm not going to let go. If you mess up the configuration, it can be hard sometimes to get back to it. So in principle, you can grab that and put it on some other screen. Maybe you have more monitors or something. You can move things around however you want. And what are these views? Well, there are two lists. There's a big one. So this is just the list of all the clusters. And there's a small one. There's the similarity view. So whenever I'm clicked on a neuron on my list, and so if I'm doing manual creation, then I want to go through my list and label things as good or bad or whatever, inspect them closely. But I'm going one neuron at a time. So for this one neuron that I have selected here, has ID 359. If I press space, it gets me into this, basically it starts selecting other neurons that are close to it that have a chance of interacting with it, either because they're pieces of the same neuron, or because, I don't know, maybe they weren't cut with each other quite right, but they have very similar waveforms, right? So these are similar waveform clusters that might or might not be the same neuron, and that's what we're trying to decide when we do this manual curation. So if I press space continuously, now if I make this bigger, okay, you can see there's a lot of information here. How about, I'm going to take this out, and I'm going to expand this view so we can see it. Okay, so you see this list is sorted, this little triangle tells us that the list is sorted by similarity. And so the biggest similarity is 0.14. This is just a correlation between templates, except for some special cases we'll go through later. But it's basically the correlation between templates. And so if the biggest correlation is 0.14, it's very unlikely that these really are the same neurons, right? We found a template that was well isolated from all these other guys. And we could just kind of go through. If I press space again, it will go to the next one. But that's going to be even less similar to my original neuron. So there's no chance it's part of the same neuron. Okay, so at least now what we know is that this one cluster is very well separated from the other clusters. But there's still the possibility that this one cluster is a merge of multiple neurons. So how do we check that it's not a merge of multiple neurons? Any ideas? Say again? Right, and I heard ISI over there. Right, so that's here, the Correlogram view. Now I'm just going to click back on this one neuron so we just have it alone. So this is the autocorrelogram of this neuron, spikes with itself. So if you're not very familiar with this, all that's happening is we're going through our list of spikes from this neuron, and we are histogramming, ISIs from this spike to the next one to the next one. And there should not be any very small ISIs because we know the neurons have refractory periods. They cannot fire again after they've just fired. So if you see things, beautiful things like this that kind of collapse to 0 in the middle here, maybe there's like one outlier sample here or something. Yeah. That's good. That means you have a very good refractory period neuron. And there's another view that is kind of an extension of that one. It's a more new view in Phi 2 called the ISI view. That's really, the same, but it's done on a logarithmic scale. So what's happening here in the ISI view is we just have 1/2 of this correlogram. Okay, not quite that. It's a distribution of just the ISIs. Okay, so there's one little difference. Is when you compute ISIs, right, inter-spike intervals, it's always between consecutive spikes. Whereas when you compute autocorrelograms, it's between any two pairs of spikes. Whether they're consecutive or if there's a spike in between, it doesn't matter. You're always doing these histograms. And so these should tend to look like they're empty in the middle here, and then they gradually rise up to some constant baseline rate of kind of coincidence between two spikes. Unless you've got things that are kind of more bursty. So maybe a neuron, it can't fire 1 millisecond after it just fired, but it might want to be bursty and fire like 10 milliseconds, every 10 milliseconds or so. They will result in a particular structure in the Correlogram view that you'll learn to recognize easily. So let's start going through them. Okay, so now let's just focus on the autocorrelogram for now. I'm just going to scroll through pretty quickly, unless I find something interesting, and then we'll stop. So here's the pretty bursty one. It has this kind of burst at exactly this, probably like 2 millisecond interval. So it tends to almost always fire once more after it just fired.
Where do we see the scale of this? How do you know that's?
It's like this 25 here tells you the milliseconds.
From where to where?
From 0 to 25,
Okay.
0 in the center.
Yeah.
Right, so
What
this...
is the ISI you giving you? What are you voting? That's different.
Because it's on a log scale, you can see small numbers of ISI violations a little more clearly.
Yeah, except that I don't think it is right here, but maybe we can change that. So let's see. That's a new view. I don't know much about it. ISI view. Increase, decrease, reset scaling, set and pins. No, I don't see. Well, you can take a screenshot of it. That's good.
What would you expect to see if there were multiple neurons firing in the ISI view?
Okay. If there, so really, okay, forget about that one for a second. That's who knows what that's for. We'll figure out a use for it. The Correlogram view is the one you really want to look at.
Okay.
So that's.
And it would just be flat all the way across.
It would be pretty, yeah. If it's really, really flat, then it's probably not just 2 neurons that are together. It's like more of them. It's like more like multi-unit activity background. So if however this goes up to like halfway or something, that's when you know that you probably have maybe 2 neurons in there, or maybe 1 neuron and some background spikes. So those are going to be the tricky cases where you will build some sense of what you're comfortable with accepting as a good unit or not.
Is there anything you could do to take those out for that entire manual duration at that point?
Yeah, so good question. So it shouldn't, depends what you mean by manual curation. So some of these over merges we might be able to fix by kind of doing some manual curation, which will mean cutting out a little bit these clusters. Others, they just won't be possible to fix if they're at the noise floor, right? So if we're at the noise floor, there's just kind of no way to set a better threshold there. So you want to recognize which one of those 2 situations you're in and act accordingly, right? If something's at the noise floor, looks contaminated, nothing you can do about it, discard it, move along to the next one. Okay, I pressed next again to the next one, another beautiful spike with a great refractory period. So you can see that all of these that have pretty abrupt transitions, these are neurons that really want to fire right away, but they have to wait that long. And if you look at the ISI view, right, you can see this neuron really likes to have ISIs of let's see, 0.004 or something. So that would be like 4 milliseconds. Okay, let's keep going. Nice one. Nice one. Here's one that's not so nice, right? So one thing you have to also learn to recognize is this very central bin here. And this will be the case for all spike sorters. For most of them, more than for kilosort. There will be an artifactually low bin down here, because most spike sorters will not allow you to extract a spike, within 0.5 milliseconds of another spike or something like that. And it's not true in kilosort, because when kilosort goes through spikes and peels them off, then in principle it could come out and find another spike right there. But because the spikes that kilosort peels off have, basically they're a little bit smooth. So whenever you subtract out something from a region, basically there won't be anything left that correlates very well with what you just subtracted. So in practice, it still ends up not really being able to find the same spike again twice within 0.3 milliseconds or something. And it would do that whether it was for noise or for the bigger spikes, kind of irrespectively. So because of that, this bin here is kind of artifactually will always be low. So you always want to look at the next bin over. And so this next bin, or increase the timing here. So right now we have, looks like we have 1 millisecond bins. So let's change that. I'm going to go to the Correlogram view, and I'm going to set my bins from 1 millisecond to 0.1 milliseconds. Okay? And now, again, you can see this kind of complete zero in the middle. So maybe more like, now these lines are still to 25 milliseconds. Okay, these guidelines here are also new. By the way, this is phi 2, right? So Cyril is currently in progress with this. So if we find bugs and stuff, it's like because of that, but already I see it's a lot more stable than the last time I used it, so that's good. There are these new guidelines on these plots that are supposed to tell you roughly where you should expect the refractory period to be, because they're basically at plus minus 2 milliseconds here. And so, this neuron, actually, it kind of, drops down. There's some contamination maybe down here, but it's a little bit hard to tell how well isolated this neuron is. So in that case, maybe we want to look elsewhere. Now, we see that this shape is not particularly huge here, right? There's a good amount of variability on top of the shape. We can look at the waveform. It looks kind of like you'd expect. So if you press W, you're shifting between the three different views here. This one is snippets from the data of this one spike, this one neuron. The second view is the template that was used to detect this neuron, and the third view is the average of the spikes from those snippets.
That's
So you see,
over all the time?
that's no, the average is noisy. It's done over just the subset of snippets, whereas the template is the thing that Kills actually used to extract the data. The template, remember, it's actually the average of spikes detecting with this cluster, but after the processing, so after the high-pass filtering. So that's the main difference between the mean waveform of the raw data and the template in kilosort. You'll see some kind of filtering differences between them too, right? So you can see like this goes up a lot more.
Wait, but doesn't the template change over time? Sorry.
The template also does change over time. So what you're seeing here is basically the one snapshot of the template.
Data from what time?
From the very beginning, from the very first time you initialize this template, I think that's the snapshot we're seeing. We are in fact saving an entire time course of the template over time. We're still thinking of ways to visualize that in the GUI, or whether you even want to go to that level of complexity, but yeah, it might be worth it. Okay, let's look at the amplitudes down here. So here also there's some cool stuff going on, which is, you can see the guidelines and 10, 20. Remember, these are kilosort amplitudes, not millivolts.
They can show
You can make it show millivolts?
You can.
How do you do that?
If you hit, so if you go to the amplitude view, or, yeah, I think you hit 8, but I don't remember, but it cycles through showing raw amplitudes. This is slow, because it has to, I just noticed from the documentation, it's slow because it's regenerating those from the raw data. And those should now be, it's actually millivolts, so you can see that's about 200.
200 what? Oh,
Yeah,
I see.
Sorry, 200 microvolts, 0.25 millivolts. And if you hit 8 again, it shows you the amplitude, the amplitude factor that we use on the template for those samples types.
I think I only have 2 things.
Yeah, 3, there's feature.
Oh, there's feature,
Yeah, so that's the feature.
but there's template feature.
Well, he added that since I looked at it.
Okay, there's feature and template
It is a
feature.
documentation with these somewhere. I know what the problem is.
Okay.
I was interested. Oh, that's small.
But this doesn't look like a 200 microvolt
It's
amplitude.
a little noisy. I have not double checked to make sure.
This is not 200 microvolts. Maybe like something like this. No. This one maybe. All right. Well, we can double check that. Sorry, some of this stuff is new for us too. I'm going to cycle back to what I know, which is the. Well, now I'm not sure which one of the 2 it is. This stuff that I know. Hold on.
Yeah, they can show the amplitude number. Was the fact that it's used to
Okay, this one, yeah? Okay, good. All right. Good, and I guess on the side here you can see a histogram of amplitudes across time, both for the selected unit in blue and for the other units on this channel in gray. Now the firing rate view, that can also come in handy for some things. I don't think it's going to be very useful here, but if you've got a neuron that's like drifting and you lose it at some point, it will appear like the firing rate of this neuron will just collapse. And that will be obvious in the firing rate view. It will also be obvious in the bottom view here with the amplitudes, because you'll start losing dots. You just won't have as many dots. Right, so what you can see about these neurons that Susu recorded was the amplitudes are super stable over time. Right? There's none of that going back and up and down that you've seen before. Okay, and the refractory periods are still nice. Look at them. Okay, this one we were uncertain about. I don't think we've resolved that uncertainty, but we can at least start looking at it in relation to nearby neurons. Now, one thing that you might decide which way you prefer it, but I guess I like to normalize these Correlogram views. So if you go to View, Correlogram View, we can toggle normalization with N. That's not what I had in mind. It was already normalized. Okay. A bit confused then, but I guess let's see more of these. Okay. All right, so what's happening, right? So the blue and the red, they both have refractory periods by themselves, but not so much between each other, except maybe there's a little bit of something there, right? It's small, but it usually indicates there might be some trade-off of spikes between one and the other. I don't know if we can recognize that, because actually I now realize we have no feature views whatsoever. So where are those?
It's under new, yeah.
Okay, so we're going to add feature view. That just means PCA features. I'm not sure where I'm going to put it. Maybe I'm going to put it as another tab here. Okay, so these are just PCA features. By default, it's showing you 4 features, 2 from the biggest channel of this unit, 2 from the next biggest channel. And the center plot is a plot of that feature over time, right? And it's nicely stable and big, separated from the noise. And then all of the side plots are plotting one PC feature versus another PC feature. And so what you typically do is you kind of zoom into these plots. If anything here looks kind of bimodal, that's your your queue to investigate further, see if you would like to cut it or not. But this one, this looks like a single neuron. Now, usually, okay, so if I clicked again on that red neuron, the second, because we're always comparing two neurons, so we put the red cloud here too, and we can see they're nicely separated in these PC views. Okay, and there's another feature view that we're also missing. It's the template feature view. So this one we also had from before. Let's see. Okay, again I had to click on it to plot them against each other. So this is the projection of both sets of spikes, blue and red, onto template 1 from one neuron, template 2 from the other neuron. And so we want there to not be any, this shouldn't look like a continuous blob that's been cut artificially. It should look like two kind of separated things. But what you do see is there's this little square here. And what that says is that both of these have actually been kind of thresholded with respect to the noise. So they're both kind of small, and so the threshold we set, if you remember the 10-4 for the thresholds, that has cut these clouds so that we don't get too much contamination from the noise. Okay, good. So you can keep going. Mostly if we're already far away from the top of this list, so the similarity is low, and now we're not gonna really find anything else anymore. Okay, this is very, very good with phi. It's a big improvement in terms of speed too, because it used to be with these really large data sets. There's a lot of data that's being moved around and loaded and plotted, and this new version is really fast with that. So that's pretty cool.
So how is that similarity metric computed?
It's just a correlation between templates. With one exception, which is sometimes at the end of processing we split a neuron because we think it's bimodal, but they still keep the same template because we don't recompute those. Well, yeah, I don't think we recompute those. And then they stay as a similarity of exactly 1, and that tells us that these have been split in this post hoc step. So if you find that things are artificially split post hoc for some random reason, you might want to reduce the effect of that step, like increase its threshold so it doesn't happen as much. Let's see if we find any example of that.
So ks2 1 had the post hoc merge.
Yeah.
Does this automatically do that already?
A
That
version
was,
of that is happening throughout the optimization.
Because that was really good for getting rid of really big noise.
Huh.
And so you don't have to get through, with the moving stuff, there's always noise, right?
Right.
It was really good at getting rid of those, and then with all the neurons, it's easy to split those apart.
Yeah, you might be the first person that has success with that.
Really?
Yeah, it used to, it was very hard to calibrate. Like, if you calibrate it wrong, it might merge everything together into one big cluster, and then you kind of screwed. And so there was a threshold in there that was a little bit sensitive, and so that was never used very much.
See, for my recordings, it merged a lot of things together. It merged all noise together. It
Right.
really decreased the number of clusters. And it did merge all the neurons together, but then you just split those and then figured it out after.
I see. All right. Yeah, I mean, what should hopefully, that was partly because, those merges had to happen, partly because neurons are drifting and they get split into different pieces because when they drift. And those merges were supposed to try to merge them back together. So now we have many fewer of those examples because we're doing this drift tracking. And so the automated merges during the optimization now work a lot better. There were such merges even before in ks2 1. There was nothing they could do because of the drift. They just weren't effective. So now those things are a lot more effective. So it shouldn't be as much of a problem. It will still output kind of noise clusters for you, but now you get this label that, in principle, and if you don't want them, you can just discard them. But ks2 gives you a label to tell you what it thinks is MUA, and what it thinks might be real neurons. So a typical use scenario is one where you kind of sort by the label that Kilosort gives you, and you kind of just go through the good ones. And if you're kind of happy with them, you're going to be going pretty quickly through the good ones. And at some point, you're going to get to the bottom of that list where things are going to start getting noisy, etc., and you can just stop there. That would be a good way to use it. So now I sorted by good, and after sorting by good, I can then perhaps sort by, oh, it's already happening for me. So see, the number of spikes is the next thing it sorts by. So we've got all the good ones first, and then they go by number of spikes. And so here's a nice huge spike. And so what I would do is there's a, let's see if the shortcuts are the same at least. Merge, split, move best to good. Okay, so by best, he always means the thing on the left, so the selected blue cluster. And so we do Alt G, and now it's green because we said that was good. And so what I'm just going to do is I'm going to go through this list. Good. I'm looking mostly at the refractory period here. The other thing I could be looking at is if there's anything very similar close to it, then I might also press space, and I would look at the cross correlogram. And I'm looking for dips, because dips would indicate it's the same neuron. Instead, I see something that looks more like an actual monosynaptic connection. So that's kind of cool. Okay, so that's not it. This, this could be it very much, but this neuron has very few spikes. So it's going to be, yeah, okay. So maybe it's the same, but so here's what happened is this neuron has lots of spikes, 48,000, and something was probably carved up out of it. Some 800 spikes. And if you want to do curation to get those spikes too, then you could merge them. So I will do this, G. This, however, hasn't been fixed. So whenever you merge something, it kind of goes to the bottom of the list, and now you're in a completely different place. That's a little unfortunate. But let's finish curating this neuron. Okay, I don't see anything else similar to it. I see another monosynaptically connected, potentially, neuron. All right, cool. Let me go back to the top of my list. Go to the next one. Okay, and I want to see, okay, so this one actually, you see it has two things with similarity. One, that means it was split out of those. So let's see if that split was right. That doesn't look like it was. This looks like, again, it's a little hard to tell when the second cluster has so few spikes, but probably the same here. We could look at the waveforms, overlap them. So you can see why it separated them, right? There's the lack of this little bump here, but they're very similar. So let's group them together.
Can you still toggle normalization in the Correlogram view?
It is toggled actually. So see, if I don't toggle it, then it's really low. So I'm not exactly sure why this other one isn't normalized, which is what I was expecting to see, I guess. Okay, we can bother Cyril about that. Let's see, is there another option? Oh look, toggle logarithmic. No, that's not. There's a lot more options now.
Right?
Yeah?
At the bottom, the amplitude for the spike is much lower sometimes. That
Where?
was-
The red one? This one here?
Yeah.
So what about that?
What makes you think that makes it the same cell?
Well, that's not the same cell. We know that much. But
Oh, the one you merged just went away.
the one I merged went away.
Oh, okay, sorry, I got confused.
Yeah. Let's go back to the top of our list. Okay. Again, I press space a few times, and if I'm satisfied that there's nothing there to merge with, I just say good. I press space. Nope, nope. It's good. Press space. No, no, good. Okay, so for good recordings, for the best neurons, you'll be doing a lot of these steps. And so when things get harder, this process will be more involved, right? So I think that's probably something that we should. Now, Susu has given us a really good data set, so we don't have a lot of
Wait, right,
hard
was that
cases.
example of two cells that were
This one?
connected?
Well, we don't really know.
That,
This really big thing?
yeah.
Yeah, sure. I mean, that's what we think, but let's see if it could be an artifact. So that's a good thing to do. Sometimes there might be if this looks like it's way too huge of a peak to really be a connection, but who knows? Let's see. Let's investigate further. So the two waveforms, blue and red, there's a small difference between them, not very big. But again, that's what you'd expect from two cells that are very close to each other, which makes them more likely to also be connected to each other. Now their amplitudes, I guess, are the biggest difference, right? The amplitudes are very different scales here. So that's the main thing that tells us they're different cells. And this helps us quite a bit too. I mean, look at the ISI view, right? These have very different distributions of ISIs. So that's another thing that tells us they're probably different cells just because their firing statistics are so different. If they were similar cells or related, they'd have more similar ISI distributions. This doesn't really help us with
But
that.
that you could see in the Correlograms as well.
I guess yes, you can see it in the Correlogram because there's no.
I mean, in the
How do you
blue
see
and the
that?
red one.
Oh right,
Those
because
are
this
just
blue is
different
rising
shapes.
slowly,
Yeah.
and the red is kind of more constant. Yeah. Yeah. So I guess this is for those of you that know a little bit about Poisson statistics, right? When your neurons are firing spike trains basically at random times, that leads to exponential distributions of interspike intervals, which is what this red one looks more like. And it also leads to these relatively flat, except for the refractory period, these relatively flat autocorrelograms, which are basically just the probability that the neuron will fire another spike after one spike is basically constant regardless of how far in the future you're looking. There's kind of no dependence. That's what makes it a Poisson process. And if it's not a Poisson process, but it's like this blue one that seems to be a lot more likely to fire some, how many milliseconds? 50, 0, I don't know, some 25 milliseconds after it's just fired. That's less of a Poisson process, and then also the refractory, the Correlogram looks like that too. All right. And then if it's bursty, and I don't think we've seen one really yet, then it will have an even higher probability to fire right away. It will have these little shoulders, so a little bit like these shoulders, but bigger, right? So you can see a little bit of an extra shoulder here, than you'd expect, and that's from bursty properties. Okay, I'm going to keep going with my manual curation. Okay, good. So you see, I press a few times space for every neuron to make sure there's nothing else around it. Now this one you see has a little bit of some kind of artifact here in the very middle, like almost coincidental. That happens in some situations where it's very few actual samples that do that, but it happens when basically a neuron fires such a big spike that the scaling of the template is not sufficient to subtract all of it off. It comes again a second time and subtracts it off again. Those are very rare cases. They'll look a little bit like this. We just learn to deal with it, recognize that it's not a big problem. It'll just be like 5, 10 extra spikes that are doublets. Let's forget about it.
Would you be able to cut it out with PCA?
Well, the thing is that these two spikes are doublets, so their main distinguishing feature will be that they happen very soon, one after the other. So if you just, they'll almost correspond to the exact same waveform. It's just that, remember in the kilosort way of doing things, you can subtract the waveform from some place and come back and find something else there. And if you haven't quite subtracted it fully, you may come back even with the same waveform and find a little bit of stuff left over again. That usually things are calibrated so that doesn't happen. In some rare cases,